And we're talking about this really interesting mechanism on how we sense a drop in blood pressure, right? Because we want filtration to be even, and we want filtrate flow through the nephron to be consistent. What's really interesting is when I was talking about this figure yesterday, I was talking about cyclic camp on both sides, and also calcium and how these two second messengers help what is called a baroreflex occur when blood pressure decreases. And in reality, that's what they used to suggest. But in reality, I just found a couple new papers that show that calcium actually inhibits renin release. And so I'll show you that. And I was talking about COX yesterday a little bit, right, which is cyclooxygenase, and these are all listed. I tried to list them, type them out underneath the figure for you. And this particular enzyme is going to convert phospholipids, particularly a particular phospholipid that has a fatty acid tail that's called agridonic acid, which looks like this. And don't worry, you'll never have to draw it. I just do it under for you. And this is what this phospholipid looks like. And what COX-2 does is it's called cyclooxygenase. So it's going to cyclize it and add an oxygen. All right, great name. So you can see here it cyclizes the top by simply rearranging electrons, and then add some oxygens to it, right? And that gives you a molecule called prostaglandin E2. So prostaglandins are modified fatty acids that are really potent ligands or communication molecules in the immune system and inflammation. And obviously in this setup, and there's another enzyme right here that's called PGES. COX-2 converts agridonic acid into what's called PGH2. And then this enzyme here, PGES, which is prostaglandin E synthase, it converts PGH into PGE2. So these two are kind of complex together. They both work together to convert a fatty acid into a prostaglandin. And then the prostaglandin is going to travel from the macula densa over to these, what we call juxtaglomerular cells, right? Which are going to release renin. Now, it's pretty cool. There are two mechanisms. One involved with the afferent arteriole, which is called a baroreflex, and one that involves these tall macula densa cells, which they have the ability to sense tubular salt. And they do that through a variety of ways. And I just want to show you, how ugly it gets, actually. Okay, and that's what the next figure shows. So I found this last night, and I was fascinated by it, right? And so on the test, you're going to have to reproduce all of them. I'm just kidding. You're not going to have to reproduce all of them. Right? So what I try to do is highlight a couple of these pathways, right? But it's incredible that obviously, by just looking at this, that filtration rate is really important, right? For your kidneys and your body to function right. There are so many ways to stimulate renin release. And so the one in blue, I'll just point out the one in blue because yesterday I said an increase in calcium causes renin release. And there's a couple papers in the early 2000s that suggested that. But now they know that there are a whole bunch of ways to get calcium into this juxtaglomerular cell. And notice what it does. It breaks down CAM. Right? So it doesn't cause renin release. It actually inhibits renin release. And I'm not going to talk about how. All right? I'm not going to talk about all the different ways. One of them I'll point out immediately is here's the ER. Here is calcium coming out. And you know that it must be phospholipase C, right? That's activated in that process. And so there are obviously a number of ligands that can cause IP3 production using phospholipase C that can cause calcium release. And this would inhibit renin from being released into the afferent arteriole here. So here is and I love this figure because it's got a whole bunch of molecular biology and self-signaling on it. Again, I listed kind of the more important of these second messengers that I'm really interested in. But this salt sensing comes down to, and the one that I'm really interested in, is the activation of these prostaglandins, right? And so low salt, because the filtrate's moving too slow. Everybody understands that, right? The filtrate's moving too slow. There's not going to be as much sodium chloride in the filtrate because of too much reabsorption, right? Too much reabsorption. And the way that these cells sense this is through this particular co-transporter, sodium chloride potassium co-transporter, which we've talked about before. Notice you can phosphorylate it, opening it and closing it or affecting it. But if less sodium's coming in, that is going to first and foremost block this mechanism, which is the production of ATP. ATP usage is going to slow because the sodium potassium pump is not going to be as utilized as much because not as much sodium is coming in. And so as ATP is expended in any cell that uses pumps, obviously the mitochondria are going to ramp up, right, and produce more produce more ATP. But it blocks this because the pumps aren't working, and therefore, the ATP production goes down, and the byproducts of ATP are not going to prevent renin release. The other major thing here that I talked about earlier is the activation of COX, right? And there are a couple different ways to do this. And what I find absolutely fascinating is that most of the enzymes here that are stimulated during that salt sensing are kinases, right? Which you guys already know because that's what we're going to do. Kinases are on and off switches. And a lot of these kinases are these two right here, or a series of these that are called ERK and P38. All right, these are MAP kinases, right? Mitogen activated protein kinases. Those MAP kinases are incredibly important in cell signaling, and they were first isolated during division, during mitosis. That's what they're called, mitogen. Now, how this lessening or decrease in salt activates these kinases is not known. I looked all over the place. I can't find anything that they just have shown through knockout mice, which knock out genes and knock out proteins, that low salt causes the activation of this complex, these two MAP kinases, which then phosphorylate COX, right, which is a membrane-bound enzyme, and then that will cyclize those fatty acids and produce PGE2, right? And then prostaglandin can leave, right? It leaves and crosses the small space and binds to its receptor over here, EP24, which is just prostaglandin E2 receptor, or 4, because there are lots of prostaglandins. And it's a G-coupled receptor. And this is really interesting. So this is something that's more new. We've talked about adenylate cyclase, right, being in the cell membrane, right? But the evidence suggests that the adenylate cyclase that this particular alpha subunit is going to stimulate is in a vesicle. It's a vesicular adenylate cyclase. So we just moved it. So the alpha subunit travels through the cytoplasm, activates the adenylate cyclase in these vesicles, and that's going to lead to CAMP, right? And we know that whenever you increase CAMP, that's going to turn on protein kinase A, and then protein kinase A is going to phosphorylate this specific protein here that's called VAMP2, like a vampire, right? That's what that reminds me of. And these VAMP proteins are vesicular associated membrane proteins. Again, these names are listed under the figure. So you phosphorylate that protein, which allows dynein to recognize the vesicle and walk it, right, to the cell membrane and drop the renin into the afferent arteriole. And we know that we have angiotensinogen, right, traveling through the blood, and renin will cleave off a large percentage of the amino acids, giving you angiotensin 1, which is a kind of a vasoconstrictor, or at least partly vasoconstrictor. There is ACE2 also, even though this is mostly a lung enzyme. It's also found throughout the cardiovascular system that will start converting angiotensin 1 to 2 by cleaving off a couple more amino acids. And this is a really potent vasoconstrictor, which will back blood up in the afferent arteriole and increase glomerular filtration. The other thing that's activated is NOS, right? So NOS, and there's a beta and a normal NOS here, which is nitric oxide synthase. And it basically will remove nitrogen groups off amino acids and produce nitric oxide, which is a small gas that's very volatile and very hydrophobic. So it'll pass through the membrane in a couple different ways and travel over, and this is the one I told you to look up yesterday, right? Travel over to the juxtaglomerular cell and pass through the membrane and then activate a guanylate cyclase in the cytoplasm, which will turn it on, which will cause cyclic GMP production, and that can also lead to an increase in CAM. So those are kind of the mechanisms. You can ignore everything else, right? That I kind of want you to be able to just kind of work your way through. Particularly this one, right? This is the one I'm going to be probably going to ask you about. These two are interesting, but this is the one that I'll ask you about on the exam. This one's pretty interesting up here too. I won't ask you about this one, but I highlighted it because it also activates NOS. A pH change also activates this. And there's this exchanger, right? This is a sodium hydrogen antiporter, and the sodium coming in, we can use that, use these protons coming in to drive sodium out, right? But if there's no sodium to move this, it doesn't work. And then pH is going to change. pH goes up because we're not bringing hydrogens in, right? And that turns on NOS. And that's 2023. 2020. This was just described. So it's pretty interesting. But this will be the one I want you to follow, right? And so you can follow that one to guanylate cyclase and down to CAM. And that'll be the main one that I'll ask if I ask on the test. Right? The others, I just wanted to talk a little bit about. The other thing I want to show you is the baro reflex, right? Which is, I think, is really cool. So we'll also talk about it in the heart. And this is great. They really work this mechanism out. That's what this little box right here represents. And what I want you to notice is that every blood vessel, and we're going to talk about this in a few minutes when we get into if we get to it, we might talk about it tomorrow actually. We get into the cardiovascular system. Every blood vessel and every cavity in the cardiovascular system is lined with a specialized, simple squamous epithelium. And that epithelium is called endothelium. Right? Endo meaning within, thelium, blood vessels. So it lines the entire cardiovascular system. So depending on who you read, there's a lot of them, right? So old school papers suggest there are 62,000 kilometers of blood vessels, or miles, of blood vessels in your body. More conservative, more recent papers say about 11,000 miles of blood vessels in your body. The United States, from east to west, is 3,000 miles. So think about that. You can wrap it through almost 4 times right around the U.S., just your blood vessels. So there's a lot of endothelium. The endothelium is, when I learned it, was probably important in blood clotting. Back in the 90s, late 90s and the early 2000s. Now it is heavily studied because it's involved in all kinds of stuff, all kinds of blood chemistry. So one of the things it's involved with in the afferent arteriole is this baro reflex, right? It actually is the ability to sense stretch. Too much stretch, remember, too high blood pressure. Too little stretch is where we're actually going to activate this because blood pressure is too low in the afferent arteriole, which is what is going to deliver blood for filtration, right? So I'm going to blow this up for you because it's pretty cool looking. And so I pulled this off of another paper. This one's much simpler. And so this is a blow up here. Here is basically our renin cell, right? Our cell here, is what we call our juxtaglomerular cell, right? Or it's called a renin granular cell. This is the extracellular material out here between the endothelium, right, of the blood vessel and its connective tissue. And this is the cell membrane of a juxtaglomerular cell. Right? And there are integrins. Integrin B1, is the beta one, is the major one here, which is an integral protein that interacts with the cytoskeleton and the extracellular matrix, which is the connective tissue junk on the that surrounds cells. In particular, it's going to bind to the most common protein in your body, which is collagen. Right? That's what those green rods are, is collagen. Or there's another group of proteins called laminins, which are found in the basement membrane. And these two are fibrous proteins that are associated with the extracellular material. They kind of provide the glue and support between cells. And so the integrin betas, they bind to that, the collagen. They span the membrane, and then they bind to F-actin, which remember is right under the cell membrane, right? Filamentous actin. There's an actin network under every cell. And this actin network is pretty well developed in these juxtaglomerular cells. And eventually, these cells are, remember, pretty flat. Remember, they're spindle shaped, so they're pretty flat, which means the nucleus, which is quite large in the pictures I showed you, is really close to the cell membrane. The F-actin then is going to interact with a protein called nesprin, which is a cytoskeletal nuclear membrane protein that interacts with a group of proteins called SUN, which are nuclear membrane proteins, which then span the nuclear membrane, the envelope. And if you remember, it's a double membrane, right? The nuclear envelope. And I don't know if you learned this or not when in your 165 class or in another class I always teach you. There is a scaffolding inside the nucleus called the nuclear lamina. And it is a group of fibers. And the two main ones are lamina A and C, which travel all the way through and form a scaffolding in the nucleus that you can like hook or anchor chromatin into place, right? And other things. And what they think is pressure coming in is going to push the endothelial cells, right? Which is going to push against the collagen. And you're going to start this, right? That's going to pull the integrin, which is going to pull the F-actin, which then will pull the nesprin, the SUN, and then interact with the laminin proteins, forming this pulse, if you will, that's going to travel through the nucleus. And that, if it's too less or too much, can inhibit or increase transcription. And how cool is that? So this is all mechanoreception. You guys heard of that term, mechanoreception? It's like stretch or pressure. It causes the cell to do something. And so it's a group of membrane proteins and linker proteins attached to the cytoskeleton, F-actin, and the extracellular matrix, collagen. And when the stretch is too little, that allows or can be picked up somehow by what are called transcriptional factors, which will then turn on renin genes and cause renin production, right? Which is pretty cool. That's pretty cool. So that is the Barrow reflex, and Barrow is pressure reflex, and so it's associated with circulation. Right? We'll talk about some more like Barrow receptors when we get to blood pressure. We start talking about blood pressure, and so I thought that would be cool to add. And so both of these new figures are in the new lecture now. Okay, so again, I list giving this figure legend out of the book chapter that I found, and also a list of proteins. So both of those are going to cause renin release, right? So this activation of COX and this Barrow reflex, right? This Barrow reception causes renin release and renin production. There's also, remember this protein right here? I pointed this protein out. This is cyclic AMP receptor element, right? Protein here that is responsible for turning on promoters, right? And it can bind cyclic AMP, right? Or be phosphorylated by PKA, and it will turn on renin production so that you can replace the protein renin in the granular proteins in the vesicle as you release them, right? So that you can keep renin high in these cells. Now, once renin's released, we end up with angiotensin II. And the only thing you know about it right now is it's a vasoconstrictor, right? It causes thirst. It also causes the release of ADH, which we talked about earlier yesterday. But it's also going to cause the release of aldosterone from the adrenal glands, okay? And what aldosterone does, so what angiotensin II does is it travels to the adrenal glands, which sit on the kidneys, and causes the release of aldosterone. Right? And then aldosterone is going to travel to the collecting ducts. Imagine that, right? Exactly where ADH works. So there are ADH receptors there, right? And there are also aldosterone receptors. Now, the big difference is antidiuretic hormone is a protein. So it's got to bind to the surface, right? Aldosterone is a steroid. It's hydrophobic. So it doesn't need a receptor on the surface. It passes into the cell through the membrane and binds to its receptor in the cytoplasm, right? So the aldosterone receptor is actually a cytoplasmic receptor. Just remember, all your steroids are derivatives. I think I've mentioned this, but we'll see pictures of it when we get to the endocrine system. Are derivatives of cholesterol. Okay. Once it binds to its receptor, it then travels into the nucleus, where it'll be shuttled to a promoter, where it'll turn on transcription. Okay. So it turns on transcription directly. It doesn't really form any second messengers. It can go right into the nucleus. And so it turns on messenger RNA synthesis or transcription. These messenger RNAs leave and then bring about these three major physiologic changes based on the genes that are turned on. The first one is we increase sodium-potassium pumps. So we move more sodium chloride out of the cell so that we can draw more of it in from the filtrate. So this is, remember, sodium-potassium pumps, and I hope you've noticed this. Sodium potassium pumps are almost always basally located, right? Or basolaterally located. So whenever you see a drawing and you see sodium potassium pumps, you know you're on the basal side here, or the interstitial side. And then over here's the filtrate side, right? And so we're going to increase our number of pumps so that we can keep the sodium levels really low and drive it into the cells. Along with that, it makes sense we're going to add more sodium channels up here, right? And so that's called the permease hypothesis, that we increase more sodium channels. And then we increase protein synthesis for metabolism so that we can actually produce more ATP, which makes sense, so we can drive the new pumps that we're making, right? All of these three physiologic changes are going to cause the reabsorption of salt. ADH also will be released because of angiotensin II, which will then cause water to follow it, right, through the aquaporins. And that will not only increase blood volume, but it'll also increase your solute concentration, correct? That way you're not just reabsorbing water, right? You're not diluting your blood. And since I taught that couple mechanisms to you, that I just decided not to go into these mechanisms so that I don't blow your brains up. Right? Okay, so far so good? Any questions from any of that stuff? And again, I can put this lecture back online if you want to hear it again. I put the first one on Brightspace. Now, this is a pretty cool picture here. So this is a desert rodent. It's called the sand rat. And this is a semi-aquatic to like rodent that lives near water. Okay? And the major thing that you see with all rodents, there's only one renal pyramid. So you remember I showed you the human one, and we got like 5 or 6 or 7 of them. They only have one big pyramid. So cortex, pyramid, right? And their pyramids are really long. Notice that the papilla actually comes out of the kidney, whereas their papilla is really short. Right? See that? So what's the big deal with that? All of these stripes that you're seeing are loops of Henle collecting ducts. They're really long, right? So they're really, really long. So these desert rodents, like the kangaroo rat, sand rat, and several other desert rodents, have really long renal pyramids so that they can have really long juxtaglomerular nephrons. That's a cool picture. Look at this drawing. I mean, the nephron is incredibly long. So what does that mean? The longer the loop, the more sodium chloride you can remove from the filtrate and put in the medulla. The more hypertonic the medulla is, the more water you can draw into the collecting ducts, right? So instead of doing 1,400 or 4 times blood osmolarity, the sand rat and the kangaroo rat can almost do 16 times their blood osmolarity, which means the urine that they're producing is pasty. It's almost like dry. There's very little water that is in that material. So, if I ask you on a test, why would increasing the size of the loop allow a rodent to, or a new animal that you find to concentrate urine, you would be able to explain it, right? Longer countercurrent multiplier and exchanger, better ability to draw water out of the collecting ducts. Now, we've talked a lot about tubular reabsorption. And remember, you can also secrete material into the nephron, right? And so, basically, secretion is going to be controlled by excess. If you have a bunch of stuff extra in the body, you can secrete it, right, and filtrate it, so that you can get rid of anything that's really high in the blood, so that you can keep the blood clean, if you will, of waste products. That's really the definition of waste: is anything in excess that you can't use, that you're going to get rid of. So what's interesting are a lot of what they call the pumps, or these symporters and antiporters. They are non-specific, meaning they'll bind anything organic. They'll bind anything that's positive, like a cation, right? Or anything acidic or basic. And they will move it just based on concentration. So what I mean by that, if you have a whole bunch of it in the blood, after you filtered it, it can move in, right, to the filtrate. If you don't have a whole bunch of it in the blood, it's not going to move, right? So a lot of this moving hydrogens, moving urea in particular, it's just really going to be based on whether it's high in the blood capillaries after we move through the glomerulus or not, and whether you have the right pump here, right? So here's an example of controlling, for example, pH, right? So here's your tubular fluid, your filtrate, and here's your blood, right? So this is basal, and I know that because here's the sodium potassium pump. And this is apical, right, pointing out into the tube. And what we're controlling here is protons and bicarbonate, both of which can play roles in pH, right? So if you eat something that's really acidic, or you drink a lot of acidic fluid and your blood has got a lot of acid in it, you can filter that out by using carbonic anhydrous, using carbon dioxide, and then you can move those hydrogens, right? You can move those hydrogens into the tubular fluid by either using this membrane-bound carbonic anhydrous or a cytoplasmic carbonic anhydrous, which can take carbon dioxide out of the blood, make carbonic acid, break it down to bicarbonate and protons, and then you can just get rid of the protons, right? You can do that through a sodium antiporter, using the sodium concentration to pump hydrogens out, or you can just use a hydrogen. Both of those will remove acid. You do the same thing with bases. If you look right here, here's if I have very basic blood, here's the interstitial fluid, here's the filtrate. I can use carbonic anhydrous again and then get rid of bicarbonate, right? I in this instance, instead of a sodium antiporter, we're using chloride as your antiporter. In this particular example, this is one of the few examples where they actually show sodium potassium pumps in the apical membrane, which is pretty rare. But then, again, you just use carbonic anhydrase to allow bicarbonate or protons into the filtrate from the blood using an antiporter, either a sodium or chloride antiporter. Down here, this shows anything that's organic. This is an organic transporter for organic acid. That's what OA stands for. If you wanted to put OB, it would be organic base. So something that is a metabolite of metabolism, for example, that's acidic, that actually will donate protons to fluid. And we don't want those, right? We want to get rid of those. So notice here, we have blood, and we have tubular fluid, and anything that's organic, carbon hydrogen base that is a acid, or anything that is an organic base, can be moved into the tubular cell here, usually the distal convoluted tubule, proximal convoluted tubule, either hydrophobically, or it can move with a sodium transporter, right? Allowing sodium in, the OA jumps on sodium's back. That brings this in, which is obviously going to be run by a sodium potassium pump. So this would be a sodium symporter, sodium organic acid symporter. That brings the organic acid in. And then we just allow it to escape to the other side. And typically we can bring in something else. In this instance, this is an amino acid. KG is, it's an amino acid metabolite. It's the ketoglutarate, which you can use to make amino acids and other organic molecules. So there are a bunch of different apically located organic transporters that are either uniporters or symporters or sometimes antiporters that allow you to move or secrete acids and bases into the tubular fluid. A transporter like this would move antibiotics, for example, right? Because you secrete antibiotics like this. So those are a couple examples of, and I like acids and bases, and then some organic transport that you can do secretion with. Now, the last thing I'll cover in the kidney chapter is how an organism deals with nitrogenous wastes. So nitrogenous wastes occur when we break down proteins and nucleic acids. And you can imagine you have to recycle those things. You don't just always build them, but sometimes you got to break them down. And you can break them down into their monomers, which are amino acids and nitrogenous bases, and then you can remove the amino group, right? And the amino group is NH2, add a hydrogen to it and form ammonia, right? NH3. Ammonia is NH3, right here. Ammonia is incredibly toxic. I mean, really, really toxic because it is a very strong base. It increases pH really fast, which affects your enzymes. Your enzymes fall apart. They don't work right. They also, if you ingest ammonia, can cause that they oxidize so readily that they produce a lot of heat. If you were to ingest a bunch of it, you can actually burn tissue. It's kind of weird. But ammonia is actually one way I could actually get rid of my nitrogen waste, right? But ammonia is toxic. The other thing is, if you look at ammonia, it's very hydrophobic, right? It's really hydrophobic. And so, because it's so hydrophobic, if you want to rid your body of ammonia, you need a lot of water. You need a lot of water to dump it, to get rid of it, right? With kidneys or whatnot. And so it makes sense that any animal that produces ammonia, which is metabolically efficient, you don't have to do anything. You break off the ammonium group, the amino group, and it forms ammonia, and you just excrete it. But you lose water. It's the big problem. And it makes sense that, aquatic animals are going to do this, right? Fish live in water. They can drink all the water they want. right? So they can just get rid of them pretty easily. Terrestrial animals, on the other hand, that doesn't work, right? Or would dehydrate really quickly. So we want to produce something that, first and foremost, is less toxic than ammonia by itself, and also more water soluble, at least in one instance. And that is urea. So urea is probably, if you look across terrestrial organisms, it's probably the most common way that a terrestrial vertebrate, for example, or a terrestrial insect, in some cases, will deal with these amino groups. They don't want to produce ammonia, so what they do is they attach their two amino groups to a C double bonded O. And this oxygen here, right? This oxygen can hydrogen bond water, so you can dissolve in water pretty readily. The other thing that I can do is make this compound and get rid of a lot of nitrogen at once. So it's very effective. It takes a lot more energy to do this, but it allows me to remove or compact a bunch of nitrogen together so that I can produce less waste and then get rid of it, right? And this is uric acid. Now, when you look at uric acid, I know you see these oxygens, but it's a big ring. It's very hydrophobic. It's very hydrophobic. And so you might think that, oh, that's like ammonia. I'm going to have to use a lot of water to get rid of it. But in reality, I don't. So I'll talk about that in a second. So these are the three kind of ways. Urea, less toxic than ammonia and requires less water to get rid of it because it is water soluble. Uric acid is how most reptiles, most birds, most desert animals are going to deal with nitrogenous waste because they can dehydrate this and release it as guano. You guys heard of guano before? Or paste, right? Or paste. There are some exceptions to the rules here, by the way. So, for example, with uric acid, notice it says many reptiles, birds, insects, and land snails. But there are desert toads that produce uric acid. All bats produce uric acid. Right? So there are lots of exceptions to the rules. So a lot of it has to do with how much water do I need to get rid of these? That's the first driving factor. The amount of water needed to get rid of it. And then how toxic it is. Okay? So this process of removing amino groups from carbon skeletons is called deamination. Okay? So amynotelic animals, or monotelic animals here, depending on how you say it, include most aquatic organisms like teleos. So teleos, if you don't know what a teleos is, that's a bony fish. So most fish, most aquatic invertebrates are ammonia secretors. Another really good example of this, all tadpoles are ammonia secretors. You know, so frogs, you guys know frogs, they produce tadpoles first. Tadpoles live in water for a certain amount of time, right? And then they become terrestrial frogs. And in the tadpole stage, here's a tadpole right here, they produce ammonia. As a terrestrial organism, they produce urea. So during their larval stage, they produce ammonia, and during their terrestrial stage, they produce urea. That's kind of cool. Almost all ammonia in fish is released over the gills. This is true of also tadpoles if they have gills. If not, are going to let it cross through the kidneys. In some cases, in most cases with other amphibians. Now, there is one example of a mammal that can survive large amounts of ammonia. And I told you how ammonia is really toxic. Wombat from Mexico. These guys live in the desert. They produce uric acid. They produce a lot of it, and they live in caves during the day, right? And so they're dropping a lot of uric acid, which is then going to break down into ammonia, right? And that ammonia fills the cave up. And if you walk into that cave, you'll die. Right? So if a scientist wants to study them, they wear oxygen into the cave. Animals wander into the cave, they die. It's pretty incredible. And for a long time, they were trying to figure out how the heck these bats do it, because they don't have anything special. Like, for example, their kidneys don't filter ammonia better, right? They don't tolerate ammonia better in the blood. what they think is their respiratory membranes in their lungs are heavily mucus. They have a lot of mucus, and that mucus plays a role in capturing or filtering or storing ammonia. Because when they fly out of the cave, the first thing they do is breathe tons of ammonia into the air, like they're releasing it from their mucus. So they're storing it so that it never enters the blood through a mucus trap. Basically, is what they think. That's the most recent hypothesis. So pretty cool. They can tolerate up to 1,800 parts per million of ammonia in their caves, which is just incredible. Now, urotelic animals, or ureotelic, animals are going to produce urea. And almost all of them do it through this cycle, okay? And I'm not, again, you don't have to, I'm not going to draw this, okay? But the name of this cycle is the ornithine urea cycle. And the reason it's called that is because you produce urea and you start the cycle with ornithine, right? Which is an amino acid intermediate, basically. So, what's cool is part of this happens in the cytoplasm and part of it happens in the mitochondria. Because the mitochondria is a place where you can actually make a number of amino acids. So, but if you look here, we start with this big old compound here that's called citrulline. Citrulline is basically a byproduct that comes off the Krebs cycle. You know, if you remember the Krebs cycle, you're basically making glucose. And citrulline can be converted into arginine, right? Which is down here, and this is how we do it. We actually let the citrulline come off of the Krebs cycle, we bring it into the cytoplasm, and then we use a bunch of enzymes to create arginine over here. Okay? Now, what I want you to notice about arginine is if you look at its R group here, or its R group, there's its amino end, there's its carboxyl end, right? Its R group has 3 amino groups on it, doesn't it? So what we do is we just use arginase, which cuts this off and replaces one of them with a double bond oxygen. That's what arginase does. It's the major enzyme here that is needed to remove ammonia off of arginine. And then we produce ornithine, which starts this whole thing over again. And this is how we can cycle off ammonia. We can cut that off of ornithine and produce citrulline again and throw it back into the Krebs cycle. So all vertebrates that use this kind of cycle use this particular enzyme cascade to make urea. Urea is quite soluble in water. It doesn't cross membranes very well, so you need urea transporters, right? Most vertebrates, some teleosts. When I say most vertebrates, really it's most terrestrial vertebrates do it this way. And then we know a elasmobranchs. These are sharks. We know the sharks and rays, the cartilaginous fish, they release urea. And we also know they concentrate that in their blood as an osmolyte, don't they? We talked about that. Remember, a lot of elasmobranchs have a blood osmolarity very close to salt water as far as the number of solutes. And the solute that they concentrate isn't sodium chloride, it's urea. The last one are uricotelic animals. They produce uric acid. This is what uric acid, this is kind of the biochemical pathway to production of uric acid. Uric acid is very water insoluble. It's very large. And so what that means is it precipitates really well. So that's the major difference. It's a humongous molecule and it's hydrophobic. And so what these animals will do is they'll produce it, right? And then they'll dump it into the bladder or a organ that is not found in all mammals. That's called the cloaca, right? The cloaca here. The cloaca, if you haven't had any comparative anatomy, the cloaca is a common sac that the digestive tract, the urinary tract, and the reproductive tract dump into before it leaves the body. This word cloaca, actually, if you do the literal Greek translation, means sewer. That's the translation. So interestingly, birds have cloacas, reptiles have cloacas. You know most mammals don't have cloacas, but bats do. Why? Because they use uric acid. Right? They make uric acid, and it's the cloaca where they dehydrate the uric acid. So if they produce this and the kidneys filter it, right, they're going to dump it into the bladder. And in order to hold it in the bladder, they got to dump a bunch of water in the bladder. They got to fill the bladder up with a lot of water. Then they got to run that into the cloaca. And in the cloaca, it'll sit in the cloaca, and the epithelium in the cloaca absorbs all the water. And as it absorbs all the water back, the uric acid precipitates into a paste that we call guano. And then you release that guano into the environment. And so, a lot of people collect guano because look at all these nitrogen groups. It's a great fertilizer for plants.